Very much a change of gear from the last one, and I promise you there aren't any mathematical equations in my talk at all. So we've been doing this quite neat study, I think, over the last couple of years, addressing a question about what is allergy. So first of all, some definitions about what allergy is. So allergy arises from the exposure to a whole range of environmental and food proteins. So these are called allergens, and they're recognized by a branch of the immune system called IgE. And this is a very widespread clinical problem that leads to a whole bunch of conditions from allergic asthma, susceptibility to anaphylactic shock. And these are sort of things that you get. So you get hives on the skin, and then you get a whole systemic response, which results in histidine release and so a little bit of audience participation here. So how many people have an allergy or know people who have an allergy? So yeah, not pretty well 70% of the audience. So, but this is very much a developed world problem. So you don't observe this in developing countries at all. And what's the other strange thing is why are we allergic to peanuts, milk, pollen, and dust mites? So these are dust mites. So these are the most common ones. So we have some observations about this. So that branch of the immune system, the IgE branch, is thought to have evolved to provide mammals with additional rapid response to combat Metazoan parasites. So these mechanisms are the same mechanisms that you're getting when you are exposed to these different types of parasites, these helminths and arthropods. So these guys on the left here, these are schistosomal worms. These are adult males. These are pretty prevalent in sub-Saharan Africa, South America, Southeast Asia. These are the adult males. You can tell that because they have this sort of sucker region here, and this basically buries into your gut. They have quite a complicated life cycle. They go through quite a lot of stages. The female, we haven't got any females in this picture, but it sits in the groove here. And they basically sit in your gut and produce lots of eggs, and that's what causes a lot of the pathology associated with these things. But more importantly, these things live with you for about 40-odd years. So there's examples where people have acquired these infections in an affected area and then gone away, and 40 years later, they're still producing eggs in their stools. So these things are very long-lived, and you generate a very strong immune response to them when you're first exposed, which is this IgE response. And then there's some very poorly understood immunoregulation that goes on as if these things become established that downplays these responses. And the same thing happens with this, which is a mite. So this is a mite biting into your skin. Again, you have a very strong response, but continual exposure to these things in your environment, and your responses die back. So we hypothesized that basically the IgE-mediated response has evolved to provide extra protection against these Metazoan parasites rather than to give you allergy. So from that, we predict that environmental allergens will share key properties with Metazoan parasites, and can we detect this in human populations? So one of the first things we did, there's a couple of really fantastic resources that have been collated over the last few years. One's called Allergome, and the other one is IEDB over in San Diego. And basically, if you go and look at these types of databases which catalog known allergens and known epitopes, you can see that basically allergens are very much confined to the metazoans and plants, a few in fungi, and hardly any in bacteria. And this is never controversial because there's an alternative hypothesis to why you have allergy, and this is actually more to do with metagenomics of the gut. So some people have observed that if you change the bacterial content of the gut, you can remove allergy, basically. So there's this sort of kind of gave a bit more to our hypothesis because of this lack of obvious bacterial component. And we were particularly interested in 10 families that we basically culled from the literature to be associated with allergy. And these represent most of the known allergens. So these are the sort of 10 families that we've picked upon, and we kind of can divide them into 3 groups. To go without being too sort of Donald Rumsfeld about it, we have known knowns, and we have knowns and unknowns, and combinations of the above. So in our group A, we have known metazoan parasite proteins, which contain these from these families, and we know that they elicit a response with an IgE response. And there's three main families here. We have a group B, where we can detect the homologue in the metazoan parasite to the allergen, but nobody's ever tested an IgE response or seen anything like that, and that's quite a few of the families. And then we have these group Cs, where we really can't detect any obvious homology, and nobody's ever tested it as well, and that's just one family called prolamin. So just another quick aside as well about how people go about determining what an epitope is on an allergen. So most of this is actually done in sequence space, and what an experimental person will come along and do, and basically denature the protein, chop it up into sliding windows of 10, well, actually sliding windows of X length, and then test it to see if they actually have a response to a basophil assay. And you can see why I said 10, because actually this is the number they pick out. So it's a little bit artificial about what an actual length is. So you can see that 10, 15, and 20 seem to be the preferred numbers when they do the chopping up. But you can get very, very long epitope, or perceived to be long epitopes, and I'm not entirely sure how much these actually exist when you think about it. Anyway, so we developed a pipeline, which basically took two main data sets. So we had the allergenic proteins that we'd taken from Allergome and from IED databases, and we had both sequences and structures. And where we didn't have structure, we tried to model it as much as we could from known sequences. And then we had the same from a whole range of different parasites. So these were mostly from worms, from nematodes and trematodes. So this is, and then as well as a few ticks, and as much as the genome that we could model in sequence. So then we defined the domains in all these proteins, and that was through both PFAM and through Cas gene 3D. We then selected for these 10 families that we were particularly interested in and considered them for the analysis. And then we tried to identify parasitic proteins that had the same either PFAM or Cas gene 3D assignment, and either used the sequence homologues as detected by PFAM, or we tried to use the structure-based homologues as detected by Cas gene 3D. And then we basically looked for similarities based on protein sequence or 3D structure. And we did this in 2 ways. We used either FAST-A for the sequence similarities based on motifs, epitope motifs based on IEDB, or we tried to use structure and structure motifs, and we used this Binet-Cauchy search, which was specifically for a very small fragment identification. And then we did a whole lot of manual curation and analysis. So basically, I'm going to show you some results now. So we have made some predictions where which were with known Ig activity. So this is your sort of group A. So the first observation is that even though we've got we can detect the epitope, and this is what's mostly reports back from what's the literature, our overall sequence similarity is extremely low. And this really does kind of builds on Cyrus's talk from this morning, where structure is so much more conserved than sequence. And this is what's coming really to the fore here in this sort of misappropriation of the immune system. And you can see here from the alignment of our model from our model and from the known allergen protein, this is the region that is predicted to be the epitope. And so you can see that from at least at the C alpha level, it's quite well conserved. Going to our sort of B group, where we have known Ig activity, but we've made predictions where we know what we've got quite good homology, but we haven't got any unknown Ig activity. We've got another model here, and you can see that the epitope here is slightly bigger. And this is getting into sort of slightly unknown unknowns, because these are 2 models basically from the structure. And our overall sequence density is about 27%. So again, very low sequence density, but in the epitope region, quite high similarity and quite high structural similarity. And then we made some other predictions, and this is around a protein called BETV1. So BETV1 is actually found as in birch pollen, so it's the main allergen of birch pollen. And we basically managed to find a homologue, which we modeled from the schistoworm. So the first question is what the hell is a plant birch pollen protein doing in a worm? And this is even more so because, so this is function of this protein is in plant cell wall synthesis, basically. So no requirement for that so completely repurposed in this place. So we predicted what the epitope should be, and you can see that there's quite high similarity and conservation in this region. And then what we did was we took this protein, purified it, and then exposed it to people who have been infected with schistosoma. So they've got a response. And what you see here is basically, so this is a response that you expect to an antigen, and then this is the response that you get when you expose this protein to sera from infected people. So this is from people from Uganda. And you can see that there's a significant proportion of the population, so this is 220 odd people, a significant portion elicit this IgE response. So this is kind of like indicative of having a shared similarity between the allergy response and your natural response to infection from these worms. And in fact, we can delve a little bit deeper. So the response is actually a component of three things, really. You have an IgE response, and then if you have an allergy response, or you're exposed to allergy-like epitopes in your infection, you should also progress to have an IgG4 response, and then that matures into your IgE response. So this is kind of like our null experiment here. So this is what you expect to see to just antigens. So these three proteins here, we don't predict to be allergenic, but allergenic, but they are antigens. So they elicit the IgE response, but they don't progress to this IgG4 IgE. But with the BETV1, we see this progression. So, sorry, this is reversed here. IgG1, we have the response and then we get the IgG4, and we get the IgGE. And this is just going to be summarized in this here. So that kind of wraps up from this little story. So we managed to identify in total 206 parasite proteins. We were able to identify for the first time a plant protein that is a commonest allergen in pollen in a worm, and then confirmed that it was a target of IgE and schistosomiasis infected humans. And so this identification of significant similarity, inclusive of the epitope regions between allergens and Helminth proteins against which IgE is an observed marker of protective immunity, kind of explains this off-target effect of IgE-mediated immune system in allergy. So this really addresses a really outstanding question in allergy research, which is basically what makes an allergen an allergen. So I'd just like to acknowledge the postdoc who worked on this project, which is Nidhi, and our two experimental collaborators, the Dunn group in Cambridge, and particularly Edward Farnell, who did most of the experimental work that I showed, and Rick Mazel's group, who's an immunologist up in Edinburgh. And of course, you can't do this without any money, so this was mostly funded by the Wellcome Trust, and I'm funded by the MRC UK. So if you have any questions, I'm happy.
Thank you. Questions?
Thank you for the talk. Could you elaborate a little bit more how you perform this structure motif search?
Okay, so basically it's a simple scan. So we basically take the fragment and then we look for that fragment into the bigger, into the main molecule, and we see where we get the hit, basically. So this algorithm, I think, is developed by a French group. I can't remember exactly which group it was, but they were. It was very pertinent. It came out just as we were doing the study, so it was very good to be used. It seems to work.
Thank you. More questions? Let's thank the speaker again. Thank you, Nicholas.